This standard technique produces fluorescently tagged gelatin-coated cover slips that are suitable for quantitative Invadopodia matrix degradation assays. After coating the cover slips with fluorescently labeled gelatin, plate the cells and culture for matrix degradation. Next, fix and fluorescently stain the cells for Invadopodia marker proteins. Acquire the images and quantitate the matrix degradation. Typical results can quantify the differences in Invadopodia-mediated matrix degradation between cells with differential manipulation of select genes or treatment with pharmacological agents.
Visual demonstration of this method is critical as preparation of gelatin-coated cover slips with homogeneous fluorescence is difficult to achieve due to uneven coating of fluorescent gelatin or improper handling of gelatin-coated cover slips.
Demonstrating the procedure will be Research Assistant Professor Karen Martin, Postdoctoral Associate Mandy Ammer, and graduate students Elyse Walk, Karen Hayes, and Steve Markwell.
To prepare the unlabeled 5% gelatin sucrose stock solution, combine the gelatin and sucrose in PBS and dissolve completely at 37 degrees Celsius. Then store at 4 degrees Celsius. Proceed to clean the glass cover slips. Distribute an individual cover slip into each well of a 24-well plastic tissue culture plate. To each well, add 500 microliters of 20% nitric acid and incubate for 30 minutes. After removal of the nitric acid solution, wash the cover slips 3 times with